Open any biology textbook, and the Golgi apparatus is depicted the exact same way, a neat stack of flattened cisternae, packaging proteins. But that image excludes the structural reality of the organelle. These lipid discs are not floating freely in the intracellular fluid. In reality, the organelle is anchored by the Golgi matrix, a dense thicket of structural proteins reaching out to capture passing transport vesicles. The Golgi apparatus manages a continuous flux of incoming and outgoing membrane traffic. This physical matrix provides the scaffolding necessary to maintain order within that chaos. The primary components of this matrix are a family of proteins known as Golgins, defined by their highly elongated, predominantly coiled-coil structures. These proteins act as the reaching arms of the organelle. They project anywhere from 100 to 600 nanometers from the membrane into the surrounding cytoplasm. To map the Golgi life cycle, we examine membrane anchors, specific tethers like TMF1, and mitotic network dismantling. The secretory pathway is a system of biomechanical reach and capture, as much as it is a study in chemical signaling. The structural logic of a Golgin starts with a strict topological rule. Membrane attachment is localized to the extreme C-terminus. This ensures that the vesicle capturing N-terminus always projects outward. This diagram outlines 3 distinct strategies Golgins use to anchor themselves. First, giantin physically embeds a transmembrane domain through the lipid bilayer. Second, GM130 relies on intermediary protein linkages, binding directly to GRASP6-5. Third, TMF anchors thy binding directly to Rab6 at the membrane surface. These specialized anchoring strategies guarantee regional specificity, ensuring that different Golgins are segregated between the cis and trans Golgi networks. Once anchored, the reaching N-terminus is free to execute its primary function, recognizing and physically capturing its specific target vesicle. This schematic details GMAP210 intercepting transport vesicles. An ALPS motif at the tip binds high lipid curvature. The vesicle transfers down the shaft through sequential Rab binding sites, acting as handholds, pulling it close to the Golgi surface for snare fusion. Golgins act as asymmetric conveyor belts, controlling the physical pathway a vesicle takes to reach its destination. In isolated cellular assays, researchers often find that Golgins appear redundant. Multiple tethers are often capable of capturing the same class of transport vesicles in a lab setting, but genomic data for the TMF1 gene in mice reveals a different reality. Expression profiles show that TMF1 is active across 30 different tissue types, which on its own suggests a general, widely supported utility. Yet, knockout studies show that removing TMF1 does not cause a general failure across all those tissues. Instead, the absence of this single tether results in highly localized physiological defects. This suggests that while tethers may appear interchangeable in a dish, they govern specialized, irreplaceable pathways within a living organism. The first defect in TMF1 knockouts is male infertility, caused by a failure of spermatogenesis. Without TMF1, the Golgi loses the dynamic spatial orientation required for acrosome assembly. The sperm cell physically cannot mature. This failure is accompanied by a severe drop in testosterone levels and abnormalities in the epididymis. A second defect appears in the colon, where the TMF1 tether is required for the regulation of the mucosal barrier. Altered vesicle delivery in the gut changes the composition of the colonic mucus. This change reduces the tissue's susceptibility to acute colitis by reprogramming the resident microbiome. In these systems, reproductive viability and gut defense depend on the movement of individual vesicles. Cell division presents a challenge of coordination. The Golgi must be partitioned equally into 2 daughter cells. To achieve this, the cell arrests secretory trafficking, and the cisternal stack is fractured into fragments. The biochemical trigger for this fragmentation is phosphorylation by mitotic kinases. As division begins, these kinases hyperphosphorylate the coiled-coil shafts of the Golgins. This modification blocks the Golgin's ability to tether vesicles, specifically disrupting the interaction between GM130 and the docking protein P115. The result is a self-driven shift in state. Continuous vesicle budding continues, but without tethers to capture them, the cisternae dissolve into a swarm of drifting vesicles. The cell dismantles the organelle by using its own budding mechanism against it, simply by turning off the tethers. While mitotic fragmentation is reversible, the disassembly during apoptosis, programmed cell death, is permanent. In this pathway, Cas-base proteins serve as the executioners of the Golgi matrix. Instead of blocking binding sites, Cas-bases irreversibly sever the Golgin shafts, decoupling the membrane from its structural scaffold. The Golgi apparatus functions as a processing center because of the precise biomechanical reach of these Golgin fighters. It is the specificity of tethers like TMF1 that translates the movement of a single vesicle into the health and survival of an organism. The Golgi survives by being at once a rigid scaffold and a structure capable of a controlled biochemical collapse.